Welcome to the Deep Dive, where we crack open complex topics and distill them into the most essential, intriguing insights.
Great to be diving in.
Today, we're plunging into the absolutely fascinating world of DNA metabolism. When you think of DNA, you probably picture it as this incredibly stable molecule, the static blueprint of life, right?
It's the common picture, yeah.
But the truth is far, far more dynamic than that suggests.
That's spot on. While DNA is the ultimate repository of genetic information, guiding everything from the size of a cell to the shape of an entire organism, well, calling it merely stable storage really misses the bigger picture.
How so?
It just doesn't capture the constant, intricate processes by which this precious genetic information is not only preserved, kept uncorrupted, but also flawlessly transmitted from one generation to the next.
Okay, so our mission today is to unpack that complexity. We're talking DNA replication, repair, and recombination.
Three huge areas.
Exactly. And these aren't just biological footnotes. They're dynamic, relentless forces constantly at work. They ensure life's continuity, battle daily damage, and even, fuel evolution itself.
Absolutely. And the molecular mechanisms, the biochemical pathways involved, they're truly ingenious.
We'll explore those, often using landmark discoveries made with common bacteria like E. coli as our guide. These are insights you find detailed in, foundational biochemistry understanding. So let's dive in. Let's begin with DNA replication, the fundamental process of making faithful copies.
The core copying mechanism.
For centuries, long before we even knew DNA structure, scientists marveled at how organisms seemed to copy themselves perfectly. But once the double helix was discovered, the mystery truly began to unravel.
Exactly. The very structure of DNA, deciphered by Watson and Crick, it immediately hinted at the template concept, meaning each strand could serve as a guide, a template for building a new complementary strand. This idea led straight to the discovery of semi-conservative replication.
Right, the Mendelssohn and Stahl experiment.
Beautifully demonstrated by them in 1957, their elegant experiment showed that every new DNA molecule is a hybrid, one original parental strand and one brand new, newly synthesized strand. It's really a marvel of biological design.
And how does this incredible copying process actually kick off and then proceed within a living cell? Where does it start?
Replication typically begins at a unique, specific point on the DNA called an origin.
Okay.
For circular bacterial chromosomes like E. coli's, it usually proceeds in two directions simultaneously, moving away from that origin.
Two directions.
Yeah, forming two replication forks. Think of them as points where the DNA is unwinding and being copied as they move along. John Terence famously visualized this in E. coli back in the 60s.
Yeah, this structure image.
That's the one. It showed this extra loop, looked just like the Greek letter theta, confirming that bidirectional movement.
Yeah. So DNA strands run in opposite directions, anti-parallel as we call it. But new DNA can only be built in one direction, 5' to 3'. How on earth do both strands get copied at the same time? It sounds like a biological paradox.
It definitely did. It was a major puzzle. This paradox was brilliantly resolved by Reiji Okazaki.
Okazaki fragments.
Precisely. He discovered that while one new strand, the leading strand, is synthesized continuously, just following the replication fork, the other one, the lagging strand, is built discontinuously.
And pieces.
In short pieces, now known as Okazaki fragments. These are maybe 1,000 to 2,000 bases long in bacteria, shorter in us. They are then joined together later. It's like building one road smoothly and the other in short connected segments.
Before we get deeper into building DNA, what about the flip side? The enzymes that actually break down DNA, because that must play a role too, right, in managing all this.
Absolutely. Those are crucial. They're called nucleases or DNases if they're specific to DNA.
And there are different types.
Yeah, two main types. Exonucleases, which sort of chew away at DNA from its ends, either the 5' or 3' end, and then endonucleases, which can cut the DNA strand at specific internal sites, breaking it into smaller fragments. Both are critical for repair, recombination, and even parts of replication itself.
Okay. Now, for the true workhorses of DNA synthesis, the DNA polymerases. Arthur Kornberg's pioneering work in 1955 really put them on the map. So how do these molecular machines actually add new pieces to the DNA chain? What's the chemistry?
At their core, all DNA polymerases perform a fundamental chemical reaction. They take a building block, a deoxynucleotide triphosphate, or DNTP.
A's, T's, C's, and G's.
Exactly. And they attach it to the growing DNA strand. Think of it like snapping a new Lego brick onto an existing structure. The existing strand provides a little hook chemically. It's a 3' hydroxyl group.
Okay.
And the polymerase brings in the new DNTP, aligns it with the template, releases some energy from it by breaking off 2 phosphate groups, and forms that new phosphodiesterase bond, linking it into the chain.
And it needs help doing that.
Well, the reaction is stabilized by magnesium ions at the enzyme's active site. And critically, every DNA polymerase needs 2 things to get started.
But sure.
A template DNA strand to read, that's the guide for which base comes next, and a primer.
A primer, like painting.
Sort of. It's a short pre-existing piece of DNA, or often RNA, that provides that essential 3' hydroxyl hook for the polymerase to start adding onto. It can't start from scratch on a bare template.
Ah, okay. And they stick around for a while.
That's called processivity. It describes how many new nucleotides they can add before letting go of the DNA. Some are highly processive, adding thousands or even hundreds of thousands of bases.
Replication needs to be incredibly accurate.
Okay.
Considering the billions of bases copied, how does the cell achieve such amazing precision? Fewer errors than you'd expect by chance.
It's really a marvel of multiple layered mechanisms. It's not just one thing.
Like what?
First, the polymerase enzyme itself is quite picky. During that nucleotide addition step, it actively checks the shape of the incoming base. Yeah, the geometry of the correct AUT and GA base pairs is very specific. If an incorrect nucleotide tries to fit into the active site, it just doesn't have the right shape, the right geometry, and it's usually rejected before the chemical bond is even formed.
So a physical check first.
Right, but mistakes still happen occasionally. So layer 2. Many DNA polymerases have a built-in proofreading function.
Like spell check.
Exactly like spell check. It's a separate enzymatic activity, usually a 3' to 5' exonuclease. If the polymerase accidentally adds the wrong nucleotide, this proofreading nucleus detects the mismatch, pauses, backs up, and specifically removes that incorrectly paired base. Then the polymerase gets another chance to add the right one.
That must cost energy.
It does. It's energetically costly for the cell, using up extra energy bonds, but it dramatically boosts accuracy, maybe 100 to 1,000-fold. Maintaining perfect DNA copies is just that important.
What's fascinating is that even a relatively simple organism like E. coli doesn't just have one DNA polymerase. It has several, right? Each with distinct roles, like different tools in a toolkit.
Indeed. It's not a one-size-fits-all situation. DNA polymerase, or Pol-1, was the first one discovered by Kornberg. It's abundant, and it does have a unique ability, a 5' to 3' exonuclease activity that's useful for removing RNA primers and filling in gaps later.
But it's not the main replicator.
No, it's actually too slow, and its processivity is too low. It falls off the DNA too easily to copy the whole chromosome efficiently. That central heavy lifting role belongs to DNA polymerase III, or Pol-3.
And Pol-3 is different how?
Pol-3 is the real speed demon. It's much, much faster and incredibly processive. It can add hundreds of thousands, even millions of nucleotides without dissociating from the DNA template.
Wow. How does it manage that?
It's because Pol-3 is actually a huge, complex holoenzyme. Think of it less like a single tool and more like a sophisticated molecular machine made of many different protein subunits. It has the core polymerase parts, the proofreading exonuclease, but also other crucial components. One key part is a donut-shaped protein complex called the sliding clamp.
A clamp?
Yeah, the beta subunit in E. coli. It encircles the DNA like a ring and tethers the pull-through-core enzyme to the template. This prevents the polymerase from falling off, which is what gives it that amazing processivity. There's also a clamp loader complex that uses ATP energy to open and close this clamp around the DNA at the right time.
That makes perfect sense. So DNA replication isn't just a few isolated enzymes bumping around. It sounds like a precisely coordinated assembly line, a whole factory of molecular machines.
Absolutely. That's a great analogy. The entire collection of proteins working together at the replication fork is called the replisome.
The replisome.
In E. coli, it's a massive team effort involving 20 or more different proteins, beyond the DNA polymerases we've mentioned.
What else is in there?
You have enzymes called helicases, like DNAB in E. coli. Their job is to unwind the DNA double helix ahead of the polymerase, like unzipping a zipper. And they use the energy currency of the cell, ATP, to do this.
Unzipping must cause tangles, right? like twisting a rope.
It does. That unwinding introduces torsional stress, or supercoiling, into the DNA ahead of the fork. So you need topoisomerases, like DNA gyrase in E. coli, which act like swivel points. They cut the DNA, let it unwind, and then reseal it to relieve that stress.
Clever. What else?
Once the strands are separated, they're vulnerable. So single-stranded DNA binding proteins, or SSB, coat the exposed single strands to keep them from snapping back together or getting damaged.
Protecting the template.
Exactly. And remember that primer requirement. An enzyme called primus, DNAG in E. coli, synthesizes those short RNA primers that DNA polymerase needs to get started, especially on the lagging strand.
Okay, so polymerase extends, helicase unwinds, topoisomerase relieves stress, SSB protects, primus starts. Anything else?
Yes, finally. After the RNA primers are removed and the gaps filled with DNA, there are still little nicks or breaks in the sugar phosphate backbone. DNA ligases are the molecular glue. They use energy, often from ATP or NAD plus a day, to form the final phosphodiester bond and seal those nicks, creating a continuous, intact DNA strand.
It's a beautifully orchestrated dance of molecules. Truly incredible coordination.
It really is.
Let's trace the actual steps of replication in E. coli then. Initiation, elongation, termination. How does this intricate process actually unfold from start to finish?
Okay, let's break it down. Initiation is all about getting started at the right place and time. It begins at that specific origin site, or C, in E. coli.
The starting line.
Right. Key initiator proteins, like DMA in E. coli, recognize and bind to specific sequences within ORIS. This binding, fueled by ATP, causes the DNA to wrap around the proteins and forces the strands to separate in a nearby region that's rich in AT base pairs, which are easier to pull apart.
The replication bubble.
Exactly. Once that bubble is open, other proteins load the main helicase, DNAB, onto each of the separated strands. The helicase then starts unwinding the DNA in both directions, creating those two replication forks. SSB proteins jump on the single strands, gyrase gets busy relieving the supercoiling ahead, and the whole process is very tightly regulated.
Regulated how?
To make sure replication only happens once per cell cycle. Cells don't want to accidentally copy their DNA multiple times. Mechanisms involving protein binding, ATP hydrolysis, and even chemical modifications like DNA methylation ensure it's a once-and-done event each generation.
Okay, so initiation sets the stage, then comes elongation, the main copying part.
Right. This is where the bulk of the DNA synthesis happens at the replication forks. The DNA helicase continues to power ahead, unwinding the DNA. On the leading strand, DNA pole third, clamped on, just follows right behind the helicase, synthesizing DNA continuously.
Smooth sailing there.
Pretty much, but the lagging strand is the tricky one. Because it runs in the opposite direction, it has to be synthesized backwards relative to the fork movement.
The Okazaki fragments again.
Yep. Primus periodically hops onto the lagging strand template associating with the helicase and lays down a short RNA primer. Then, a polthoric complex clamps on and synthesizes an Okazaki fragment, extending from that primer until it hits the primer of the previous fragment.
How does a cell coordinate both strands being made at the same fork?
That's where the trombone model comes in. It's thought that the lagging strand template is looped out so that the pole thread enzyme working on it can physically stay associated with the pole thread working on the leading strand, even though they're moving on templates oriented in opposite directions. It keeps the whole replisome together as a single coordinated unit.
Like a trombone slide adjusting the loop length.
Exactly. As one Okazaki fragment finishes, the polymerase releases the loop. The clamplator puts a new clamp near the newly made primer. The polymerase re-engages, and a new loop forms for the next fragment. It's quite elegant.
And then cleanup.
Then cleanup. The RNA primers have to go. In E. coli, DNA poli often does this using its 5-3-minute exonucleate activity, removing the RNA ahead of it while simultaneously filling the gap with DNA. Or another enzyme, RNase H, can remove most of the RNA. Then poli fills any remaining gap, and finally, DNA ligase seals the nick between the fragments.
Okay, initiation, elongation leads to termination. How does it stop?
In E. coli's circular chromosome, the two replication forks travel around the circle until they meet, roughly opposite the origin. There's a specific terminus region containing special DNA sequences called tersites.
Roadblocks.
Sort of. A protein called TUS binds to these tersites. The TUSTER complex acts like a one-way gate. It lets a replication fork pass through in one direction, but blocks it if it approaches from the other direction. This ensures the two forks meet within the defined terminus region.
And then?
The last few hundred base pairs are replicated, and you're left with two complete newly synthesized circular DNA molecules. but they're often interlinked, like two rings of a chain. They're catenated.
Tangled up.
Yep. So a final enzyme, topoisomerase the fifth in E. coli, which is a type 2 topoisomerase, makes a transient double-strand break in one circle, passes the other circle through the break, and then reseals it. This decatenates the chromosomes, separating them so they can be segregated into the two daughter cells during cell division.
Phew. That's quite the process in bacteria. So, if that's the bacterial story, what does this all mean for eukaryotic cells like our own? How do these fundamental principles scale up? Our genomes are huge.
Right. Eukaryotic replication shares many of the fundamental principles: semi-conservative, origins, forks, leading lighting strands, polymerases, helicases, ligases. The basic chemistry is the same. But yes, the complexity ramps up significantly.
Because of size and structure.
Exactly. Our DNA molecules are vastly larger linear chromosomes, not circles, and they're packaged tightly into chromatin with histone proteins. This presents extra challenges.
So multiple starting points.
Absolutely crucial. Instead of a single origin, human chromosomes have tens of thousands of origins of replication, maybe 30,000 to 50,000 across the entire genome.
Why so many?
Because eukaryotic replication forks move much slower than bacterial ones, only about 50 nucleotides per second compared to maybe 1,000 in E. coli. If we only had one origin per chromosome, it would take weeks or months to copy our DNA. So multiple origins are essential to replicate the entire genome within the S phase of the cell cycle, which takes several hours.
And controlling all those origins must be complex.
Extremely. There's a very intricate licensing system involving proteins like cyclins and cyclin-dependent kinases, CDKs. This ensures that each origin is activated or fires only once per cell cycle. It prevents disastrous re-replication. The key helicase, the MCM complex, is loaded onto origins early on, but only activated later when the cell is ready to divide.
Different tools too, different polymerases.
Yes, eukaryotes have a larger cast of DNA polymerases for nuclear replication. The main workhorses are pol-epsilon, thought to synthesize the leading strand, and pol-delta for the lagging strand. Both of these have proofreading activity. There's also pol-alpha, which works with primus to make the RNA primer and then adds a short stretch of DNA, but it lacks proofreading.
Do we have a sliding clamp?
We do. It's called pCNA, proliferating cell nuclear antigen. It serves the same function as E. coli's beta clamp, encircling the DNA and tethering the polymerases, pol-delta and pol-epsilon, to enhance their processivity. Looks structurally different, but does the same job.
So, similar principles, but scaled up and with more layers of regulation and specialized players.
That's a good summary. More origins, slower forks, complex licensing, different polymerases, but the core logic remains.
And on a more personal level, what's a fascinating application of this knowledge that directly impacts human health? You mentioned viruses earlier.
Right. Many DNA viruses, like herpes simplex virus, which causes cold sores and genital herpes, are quite self-sufficient. They encode their own DNA polymerases to replicate their viral genomes inside our cells.
Making them a target.
Exactly. Because the viral polymerase is different from our human polymerases, it makes an excellent drug target. A cyclover is a classic example.
How does it work?
It's clever. A cyclover is a modified version of a DNA building block. A viral enzyme, one that our cells don't have, adds the first phosphate group to a cyclover, activating it. Our own cellular enzymes then add more phosphates. This activated form inhibits the viral DNA polymerase much more strongly than it inhibits our own polymerases.
So it's selective.
Highly selective. And there's more. Because of its chemical structure, a cyclover lacks the proper 3' hydroxyl group needed to add the next nucleotide. So once it gets incorporated into the growing viral DNA chain,
It stops everything.
it acts as a chain terminator. It halts further elongation of that viral DNA strand. So a cyclover hits viral replication at multiple steps, selective activation and chain termination. Very effective.
Incredible. Okay, let's shift gears. DNA might be the blueprint, but as you said, it's constantly under attack. Thousands of tiny lesions, damages can accumulate daily in just one of our cells. How on earth does life manage to maintain genomic integrity against such a constant barrage?
It's truly astonishing, isn't it? Despite this relentless assault from things like UV radiation, chemical mutagens, even just spontaneous chemical reactions within the cell, fewer than 1 in 1,000 of those initial DNA lesions actually become a permanent mutation.
1 in 1,000.
Thanks to an elaborate, multi-layered, incredibly efficient set of DNA repair systems. These systems are constantly scanning the DNA, finding damage, and fixing it.
And their importance is huge.
Paramount. The link between unrepaired DNA damage, the accumulation of mutations, and the development of cancer is incredibly strong. Think about the Ames test, which measures a chemical's potential to cause mutations. Over 90% of known carcinogens test positive as mutagens.
Wow.
And genetic defects in DNA repair genes themselves are often devastating. Conditions like xeroderma pigmentosum or inherited mutations in BRCA1 or BRCA2 genes linked to breast and ovarian cancers, these dramatically increase cancer susceptibility because the cell's ability to fix DNA damage is compromised. It really shows the immense energy the cell invests in repair. Maintaining genetic integrity is a top priority.
So what are some of these critical repair systems? Let's start with correcting those rare errors left behind after replication, the ones that even proofreading missed.
Right, the mismatches. That's handled by mismatch repair, or MMR. This system acts like a final quality control check after replication, boosting the overall fidelity by another 100 to 1,000-fold.
How does it know which strand is wrong, the new one or the old template?
That's the clever part, especially in E. coli. For a short time after replication, the newly synthesized strand is chemically distinct from the older template strand. The template strand has specific methylation tags added to certain DNA sequences, GATC sequences. The new strand hasn't been methylated yet.
So you can tell them apart?
Exactly. The MMR system, involving proteins like MUTs, MUTL, and MUT in E. coli, recognizes the mismatch and specifically targets the unmethylated new strand for repair. MUT makes a cut in the new strand near a hemimethylated GATC site. Then an exonuclease chews away a segment of that new strand, including the mismatch.
And then
DNA pol 3 comes back in to fill the gap correctly, using the methylated template as a guide, and DNA ligase seals the final nick. It can be quite costly, sometimes removing over 1,000 base pairs just to fix one mismatch, but accuracy demands it.
and in humans.
Eukaryotic cells have very similar MUTs and MUTL homologs, showing the system is highly conserved. Defects in these cause hereditary non-polyposis colon cancer, HNPCC, or Lynch syndrome. However, the exact mechanism for identifying the new strand in eukaryotes is still being worked out. It doesn't seem to rely on the same GATC methylation system as E. coli.
Okay, so MMR fixes replication errors. What about more common types of damage that happen spontaneously, like a base changing its chemical identity?
Yes, like cytosine deamination. Cytosine can spontaneously lose an amino group and turn into uracil, a base normally found only in RNA. This is a very common lesion.
And the cell fixes it how?
With base excision repair, or BER. This pathway deals with damage to single bases. The first step is a specialized enzyme called a DNA glycosylase. There are different glycosylases specific for different types of damaged bases.
Like a uracil glycosylase.
Exactly. Uracil glycosylase finds uracil in DNA, recognizes it shouldn't be there, and cleaves the bond linking the uracil base to the DNA sugar phosphate backbone, simply removing the base itself.
Leaving a hole.
Yes, it creates what's called an AP site opurinic or a pyrimidinic, a sugar with no base attached. This AP site is then recognized by another enzyme, an AP endonuclease, which cuts the DNA backbone next to the site.
Okay.
Then a short patch of DNA around the cut is removed. DNA polymerase, often pol-I in E. coli or specialized polymerases like pol-beta in eukaryotes, fills in the correct nucleotide using the opposite strand as a template, and DNA ligase seals the final nick.
And that's why DNA uses thymine T instead of uracil U.
Precisely. If DNA naturally contained uracil, the cell wouldn't be able to distinguish a correct uracil from one that arose from cytosine deamination. By using thymine, which is essentially methylated uracil, any uracil found in DNA is immediately recognized as damage arrived from cytosine and targeted for removal by BER. Very smart design.
It really is. Now, what about bigger problems? Those bulky distortions in the DNA helix may be caused by UV light creating pyrimidine dimers or chemical adducts from things like cigarette smoke.
Those are handled by a different, more versatile system called nucleotide excision repair, or NER. This system is absolutely vital for survival, especially for damage caused by environmental mutagens.
How does NER work?
It recognizes distortions in the DNA helix rather than specific damaged bases. A large multi-subunit enzyme complex called an exonuclease is recruited. This complex makes two cuts in the damaged DNA strand, one on each side of the bulky lesion.
Cutting out a whole chunk.
Exactly. In E. coli, the ABC exonuclease removes a fragment that's typically 12 or 13 nucleotides long. In humans, the NER machinery removes a larger chunk, usually 27 to 29 nucleotides long, containing the damage.
And then fill and seal.
Yep. The resulting gap is filled by a DNA polymerase, pol-I in E. coli, pol-delta or epsilon in humans, using the undamaged strand as a template, and the remaining nick is sealed by DNA ligase.
And defects here cause
Genetic defects in human NER genes are the cause of xeroderma pigmentosum, XP. Individuals with XP are extremely sensitive to sunlight because they can't repair the pyrimidine dimers caused by UV radiation. They have a dramatically increased risk of skin cancer. It highlights just how critical NER is for protecting us from environmental DNA damage.
Are there any repair mechanisms that are more direct, that don't involve cutting out and resynthesizing a whole segment? That seems like a more efficient approach if possible.
Yes, there are a few fascinating examples of direct repair where the enzyme directly reverses the damage in a single step.
Like what?
One example is enzymes called DNA photolysis. These enzymes can recognize UV-induced pyrimidine dimers, bind to them, and then use the energy from visible light to directly break the abnormal bonds forming the dimer, restoring the original pyrimidines. It's a direct chemical reversal.
But we don't have those.
Strangely, no. Photolyses are found in bacteria, fungi, plants, and many animals, but they seem to have been lost during the evolution of placental mammals, including humans. We rely entirely on NER to fix UV damage.
Interesting. Any other direct repair examples?
Another striking one is the repair of a highly mutagenic gene called O6-methylguanine. This happens when guanine gets inappropriately methylated. O6-methylguanine tends to pair with thymine instead of cytosine during replication, leading to mutations.
How's it fixed?
There's a protein called O6-methylguanine DNA methyltransferase. It finds the O6-methylguanine, binds to it, and directly transfers the harmful methyl group from the guanine base onto one of its own cysteine amino acid residues within the protein.
So the protein takes the hit.
Exactly. And here's the truly remarkable part. This transfer permanently inactivates the methyltransferase protein. It's a suicide mission. The protein sacrifices itself to repair just one lesion.
Wow. That really underscores the priority.
It absolutely does. It illustrates the immense cellular priority given to maintaining DNA integrity. The cell is willing to synthesize an entire protein just to fix one specific dangerous lesion, and then the protein is gone.
Incredible. What happens when the damage is really severe, so extensive that maybe the complementary strand is also compromised? Or there's a break, and the repair systems we've talked about can't use a template. How does the cell recover then?
That's when things get really tricky, and the cell might have to resort to what's often called a desperation strategy. This involves error-prone translesion DNA synthesis, or TLS.
Translesion meaning across the lesion.
Precisely. Imagine a replication fork moving along the DNA, and it suddenly encounters an unrepaired lesion, like a pyrimidine dimer or a bulky adduct that the main replicative polymerase, like pol III or pol delta epsilon, simply cannot read past. It stalls the fork.
Which is bad.
Very bad. A stall replication fork can collapse and lead to chromosome breaks and cell death. So under these conditions, especially if the damage is widespread, triggering what's called the SOS response in bacteria, the cell can bring in specialized TLS DNA polymerases.
Specialized how?
These TLS polymerases, like pol-V in E. coli or pol-eta, iota, kappa, zeta in humans, have much more open, flexible active sites. They're essentially designed to tolerate weird shapes in the DNA template. They can synthesize DNA past the lesion, effectively bypassing it and allowing replication to continue.
But at a cost.
Yes, a significant cost.
Yeah.
Fidelity. Because they're less picky and often lack proofreading activity, TLS polymerases frequently insert the wrong nucleotide opposite the lesion, or even opposite undamaged bases nearby. They introduce mutations.
So it fixes the stall but causes mutations. Seems counterintuitive.
It's a trade-off. From the cell's perspective, getting replication completed, even with a few errors, might be preferable to letting the fork collapse entirely, which could be lethal. It's a gamble allowing some cells in a population to survive an otherwise insurmountable barrier, even if it means introducing genetic variation. Some argue this provides fuel for evolution, generating diversity under stress.
The necessary evil, perhaps.
In some situations, yes. Though it's worth noting that some TLS polymerases are better than others. For instance, human paleta is actually quite good at inserting the correct adenines opposite UV-induced thymine dimers, minimizing mutations from that specific lesion. So it's nuanced.
Okay, so far we've covered copying DNA replication, fixing DNA repair. But DNA isn't always static. It also undergoes incredible rearrangements, a process called genetic recombination. Barbara McClintock famously discovered jumping genes, transposons, in maize way back in the 1940s, showing us just how dynamic genomes can be.
Absolutely. Genetic recombination is the third major aspect of DNA metabolism, involving the exchange or rearrangement of genetic information, either between DNA molecules or within the same molecule. It broadly falls into three main classes.
For sure.
Homologous genetic recombination, which involves exchange between DNA molecules with extensive sequence similarity. Then there's site-specific recombination, which occurs only at specific defined DNA sequences. And finally, DNA transposition, which involves those jumping genes or transposable elements moving to new locations.
And these all have different jobs.
They do. Recombination plays crucial roles in specialized DNA repair pathways, helps rescue stalled replication forks, can regulate gene expression, is vital for generating genetic diversity during sexual reproduction, and is even involved in programmed rearrangements during development, like in our immune system.
Let's tackle homologous recombination first. You mentioned repair. How does it work in bacteria?
In bacteria, homologous recombination is primarily a really important DNA repair process, often called recombinational DNA repair. Its main job is to help reconstruct replication forks that have stalled or collapsed, often at sites of DNA damage like nicks or gaps.
How does it fix a broken fork?
When a fork collapses, it can often lead to a dangerous double-strand break. The cell needs to fix this break and restart replication. Homologous recombination provides a way. Enzymes first process the broken DNA end, typically chewing back the 5' ending strand to create a 3' single-stranded tail. This 3' tail is then coated by a key bacterial recombinase protein called RIAA. RIAA forms a helical filament on this single-stranded DNA. This RIAA filament is amazing. It can search the entire genome for a matching homologous sequence on an intact DNA molecule, like the sister chromosome.
Finds the backup copy.
Exactly. Once it finds homology, the RIAA filament promotes strand invasion, where the single-stranded tail displaces one strand of the intact duplex and base pairs with the other. This creates a branched DNA structure.
Sort of a crossover point.
Sort of. It sets the stage. This branch point can then move along the DNA, branch migration, potentially forming a cross-shaped structure called a holiday intermediate or holiday junction. Specialized enzymes then cleave this junction in specific ways, resolution, and DNA ligase seals any remaining nicks. The end result is that the broken DNA is repaired using the intact molecule as a template, the replication fork is effectively reconstructed, and DNA synthesis can restart.
So, in bacteria, homologous recombination is fundamentally intertwined with replication, rescue, and repair.
Primarily, yes. It's a vital survival mechanism.
But in eukaryotes, like us, it plays a critical role in a very different but equally fundamental process, right? Meiosis.
Indeed. In eukaryotes, homologous recombination is absolutely essential for the precisely. Without these chiasmata formed by recombination, homologous chromosomes often fail to segregate properly, leading to errors. Plus, this crossing over has another huge consequence. It shuffles the genetic alleles between the maternal and paternal chromosomes, significantly increasing the genetic diversity in the resulting gametes.
And if this process fails?
The consequences can be severe. Failure of meiotic recombination or improper chromosome segregation is a leading cause of aneuploidy gametes and resulting embryos having the wrong number of chromosomes. Trisomy 21, causing Down syndrome, is a well-known example. Aneuploidy is a major cause of pregnancy loss and developmental disabilities in humans.
And this relates to maternal age.
Yes, the increased risk of aneuploidy with advancing maternal age is thought to be linked, at least in part, to the incredibly long time human eggs remain arrested in meiosis, the dictyat stage. Those chiasmata formed early in fetal development have to remain intact for decades, potentially becoming less stable over time, increasing the chance of segregation errors later in life.
Fascinating. You mentioned proteins detecting DNA damage earlier. Thinking about double-strand breaks, how does the cell first sense that kind of serious damage in our complex chromosomes?
That's a great question. One of the key first responders to DNA breaks, especially single-strand breaks, but also involved near double-strand breaks, is a protein called PARP1 that stands for poly-ADP ribose polymerase 1.
What does PAP1 do?
PRP1 is constantly scanning the DNA. When it encounters a break, it binds tightly to the broken ends. Upon binding, it gets activated and starts synthesizing long branched chains of a molecule called poly-ADP ribose, using NAD+ as a substrate. It attaches these polymers to itself and other nearby proteins,
Creating a signal flare.
Exactly. This burst of poly-ADP ribose polymer acts like a molecular flag or signal flare at the site of damage. It's negatively charged and bulky, and it serves to recruit a whole host of other DNA repair factors, including proteins involved in both base excision repair and double-strand break repair pathways to that specific location. It essentially shouts, "Damage here, needs fixing."
And this has therapeutic relevance.
Huge relevance now. PARP inhibitors are a major class of cancer drugs. The logic is this. Many cancers, particularly those with mutations in the BRCA1 or BRCA2 genes, already have defects in their primary pathway for repairing double-strand breaks, which relies on homologous recombination. These cancer cells become heavily dependent on other repair pathways, including those initiated by PRP1, just to survive.
So you target their backup plan.
Precisely. By inhibiting PRP1, you take away that backup. The BRCA-deficient cancer cells accumulate too much DNA damage, particularly double-strand breaks during replication, and they die. Normal cells, which still have functional BRCA pathways, are much less affected. It's a great example of exploiting a cancer's specific vulnerabilities, synthetic lethality.
Okay, so homologous recombination is great for repair if you have a template. But what if a double-strand break occurs and there's no homologous chromosome nearby to use as a template, say outside of the S or G2 phases of the cell cycle?
That's where the other major double-strand break repair pathway comes in, non-homologous end joining, or NADH. This is actually the predominant pathway for fixing double-strand breaks in mammalian somatic cells throughout most of the cell cycle.
Non-homologous meaning it doesn't need a template.
Correct. Instead of searching for a homologous sequence, NADH simply takes the two broken ends of the chromosome and essentially sticks them back together.
How does it do that?
It involves a set of core proteins. First, a protein complex called Q7280 acts like a cap, quickly recognizing and binding to the broken DNA ends, protecting them from degradation. Q then recruits other factors, including a large protein kinase called NAPKCs. These proteins help bring the two ends together. Often the broken ends aren't clean, they might have damaged bases or overhangs. So various nucleases like Artemis and specialized DNA polymerases process the ends, trimming them or filling small gaps. Finally, a dedicated DNA ligase complex, Ligastore with XRCC4, joins the processed ends back together.
But if you're trimming or adding bases randomly...
Exactly, that's the catch. NADH is considered an error-prone or mutagenic repair process. Because it doesn't use a template, the processing steps often result in small insertions or deletions of nucleotides, indols, at the site of the break. The original sequence isn't perfectly restored,
But it fixes the break.
it fixes the break, preventing catastrophic loss of chromosome fragments or cell death. For somatic cells in a large organism, losing a few base pairs might be an acceptable price to pay to maintain overall chromosome integrity. It highlights the cell's absolute need to fix double-strand breaks, even if the fix isn't perfect.
Okay, let's move to the second type of recombination. Site-specific recombination. How is this different? You said it happens only at specific sites.
Right. Unlike homologous recombination, which can happen anywhere there's sufficient sequence similarity, site-specific recombination is strictly limited to particular, usually short, DNA sequences, the recombination sites.
And it uses different enzymes.
Yes. It relies on specific enzymes called recombinases. These recombinases recognize their target DNA sequences, bind to them, and then catalyze the cutting and rejoining of the DNA strands. They essentially act as both sequence-specific endonucleases and ligases all rolled into one.
What kinds of rearrangements can it do?
Depending on the location and orientation of the recombination sites, site-specific recombination can lead to different outcomes. If the sites are on the same DNA molecule and oriented inversely, recombination causes an inversion of the DNA segment between them. If they're oriented directly, it leads to a deletion of the segment as a circle. And if the sites are on two different DNA molecules, it can result in an insertion or integration of one molecule into the other.
An example.
A great bacterial example is the XRCD system in E. coli. Sometimes, due to errors in replication or repair, the circular bacterial chromosome can end up as a dimer, two circles linked together. This prevents proper cell division. The XRCD recombinase acts at a specific site called DIF present on the chromosome. Site-specific recombination at DIF resolves the dimer back into two separate monomer circles, allowing the cell to divide successfully. It's essential for chromosome segregation.
Very precise control. Finally, the third type. The jumping genes transposons, DNA transposition.
Yes, transposable elements, or transposons. These are fascinating segments of DNA that possess the remarkable ability to jump or move from one location in the genome to another. This process is called transposition.
Yeah, it's random.
The movement itself doesn't usually require sequence homology between the transposon and its new insertion site, and the insertion site is often, though not always, relatively random. Because of this potential to disrupt genes or genome structure, transposition is generally a rare event and is often tightly regulated by the cell or the transposon itself.
What do transposons contain?
The simplest ones in bacteria, called insertion sequences, or IS elements, typically contain only the gene encoding the enzyme needed for transposition, the transposase, flanked by short inverted repeat sequences that the transposase recognizes. More complex transposons carry additional genes besides the transposase, such as genes conferring antibiotic resistance.
Ah, so that's how antibiotic resistance can spread.
It's a major mechanism. When a complex transposon carrying an antibiotic resistance gene jumps onto a plasmid, a small circular DNA molecule that can be transferred between bacteria, it allows that resistance to spread rapidly through a bacterial population via plasmid transfer. It's a huge driver of the antibiotic resistance crisis.
How does the jumping actually work? Are there different ways?
There are two main pathways. In direct transposition, sometimes called simple or cut-and-paste transposition, the transposase enzyme cuts the transposon completely out of its original location, often leaving behind a double-strand break that needs repair, and then inserts it into a new target site.
So it moves, doesn't copy itself.
Correct. The second mechanism is replicative transposition. In this case, the entire transposon is replicated during the transposition process. One copy remains at the original site, and a new copy is inserted at the target site. So the number of transposons increases.
And when they insert, they leave a footprint.
Yes, a characteristic feature of most transposition events is the duplication of a short sequence, typically 3 to 12 base pairs, at the target site. This short target site duplication ends up flanking the inserted transposon element on both sides. Finding these flanking direct repeats is often a clue that a particular DNA segment is a transposon.
Now, in vertebrates, you mentioned some DNA rearrangements are actually programmed, essential parts of development. The immune system example.
This is truly one of the most stunning examples of programmed recombination, specifically related to site-specific recombination and potentially evolved from transposition. It's how our immune system achieves its incredible diversity.
Diversity to fight off infections.
Exactly. We need to be able to produce millions, maybe billions, of different antibodies, immunoglobulins, and T-cell receptors to recognize and fight off the vast array of potential pathogens we might encounter. But we certainly don't have millions of separate genes encoding each one.
So how do we do it?
Through gene rearrangement. The genes encoding the variable regions of antibodies and T-cell receptors are assembled during the development of immune cells, B cells and T cells, from separate gene segments. For antibody light chains, for example, there are multiple variable V segments and joining J segments stored in the genome.
And they get pieced together.
Precisely. During B-cell development, specific enzymes recognize special recombination signal sequences, RSSs, that flank these V and J segments. Proteins called ARIG1 and ARIG2, recombination activating gene products, act like a site-specific recombinase. They make precise double-strand breaks at the RSSs next to one chosen V segment and one chosen J segment. Then, the cell's general DNA repair machinery, particularly the NADH pathway we discussed earlier, joins the ends of the chosen V and J segments together. The DNA between them is deleted. This creates a unique functional VJ exon that encodes part of the antibody's antigen binding site. Since there are many V and J segments to choose from, and the joining process itself can be imprecise, adding more variation, this combinatorial process generates a huge diversity of antibody genes from a limited set of starting parts.
And this looks like transposition.
The RG enzymes and the RSS sequences bear striking similarities to transposases and the ends of certain transposons. The mechanism of cutting and rejoining is also very similar. This has led to the fascinating hypothesis that the entire VDJ recombination system, the foundation of our adaptive immune system's diversity, might have actually evolved from an ancient invasion of a vertebrate ancestor's genome by a transposable element, which was then harnessed and repurposed for this vital immunological function.
Wow. Evolution repurposing molecular tools in amazing ways.
Truly remarkable.
What a journey. We've gone from the precise, almost magical copying of our genetic code in replication
Yeah, the fidelity is amazing.
to the vigilant repair systems that tirelessly guard against damage.
Constantly patching things up.
And finally, to the deliberate, sometimes programmed rearrangement of DNA through recombination. It's abundantly clear that the idea of DNA as just stable storage is, well, far too simple.
Absolutely. It's anything but static. It's a continuous, dynamic dance of incredibly sophisticated molecular machinery. This interplay underpins life itself.
This constant interaction between replication, repair, and recombination, often using shared or repurposed molecular parts like NADH and FADH recombination or recombination proteins, helping restart replication forks. It really highlights how evolution builds complexity, doesn't it? Tinkering with existing tools.
It absolutely does. It's a masterclass in molecular repurposing and integration. And it definitely raises an important, maybe profound question. What other fundamental biological processes are underpinned by such ancient repurposed molecular machinery, machinery whose origins and full connections we are yet to fully appreciate?
That is a great thought to ponder. What else is hiding in plain sight, built from old parts? Thank you so much for taking us on this deep dive into the dynamic world of DNA metabolism.
My pleasure. It's a fascinating subject.
We hope you out there feel a little more well-informed and perhaps like me, a lot more amazed by the intricate, relentless activity happening inside every single one of your cells right now.
Keep exploring, keep questioning.
Exactly. Keep exploring, keep questioning, and we'll see you next time on The Deep Dive.